So you said it's open a notes, open a book.
Yes, the exam will be open notes and open book. Um, if it was in like a classroom setting, I would probably have no open notes, not open book, and just like a note card. And I actually recommend that for the exams is that you prepare a note card of a couple of the facts that you're having a hard time remembering or like some really important information that you want to make sure that you get right on the exam. But usually like things that you're having trouble remembering so that you have that as a note card. That's usually what I do in a face-to-face course. But since proctoring, like having someone to watch you for an exam, is just such a big hassle. So it's hard to schedule that with a proctor. Are there enough proctors available? That kind of thing. And then if I don't have proctors, some students will use notes and will use book, even if they're not supposed to. So I just give it open to everybody. So I still do recommend you just prepare that one note card. So do your studying, prepare a note card, and then if you go in with just that, you'll know all the answers already. You might have to refer to it 2 or 3 times, but that's what I recommend for studying for the tests. But it will be open note and open book, but with the time limit, you're not going to be able to go in totally cold and search for every answer. You just won't have time for that. So there is that time limit on the exam. But yes, it is all the exams, including the final exam, will be open note and open book. So take good notes because it'll be easier to search through those than through a textbook if you're having trouble remembering one of the answers. And again, the exams are entirely multiple choice as well. So any other questions about the logistics of the exam? I know it's a little bit of ways away.
Sorry if you already said this, but what is the time limit for the exam? How long are they?
They're all a little bit different because some have a few more questions than others. I can go and check what this upcoming exam is, though. I'll just have to log into Salt Lake Community College. So let me do that right now, and then I can, because I think it's about 70 minutes, but it might be 70 questions in 90 minutes. So let's just double check. I should probably move this so you don't see answers yet on my shared screen there. All right, the time limit is 90 minutes for this first exam, and it is going to be 60 questions. So 60 questions in 90 minutes, so you have an hour and a half to take the exam. All right, so you can start preparing for that. One of the best ways to prepare for exams in any class, really, is to be reading your book before you go to class, so that when you go to class, you already have covered the material that we cover in, say, a lecture, either we or in any other class. So then during the lecture, you can add to your notes, like examples that are given by the professor, those kinds of things. For example, today we'll watch some videos on epigenetics, and that might help you understand it better. So adding that into your notes, and then before the exam, going and reviewing your notes. And then you can even quiz yourself after reviewing your notes. For example, I give a study guide, so you can use that study guide and kind of quiz yourself on some questions, things that you're struggling on. Then I would recommend maybe writing those on a note card. And then you could refer to that. Not all classes will let you use notes or a note card, but I think that's good practice to rewrite those notes down or rewrite those ideas down. Cognitive research shows that it will help you remember it. So even when that process of writing it down helps it get into your memory more. So then you don't even need to use that note card. It's a really neat trick. So reading before you come to class, going through lecture, adding to your notes, reviewing your notes, quizzing yourself. Those are some of the best ways to prepare for any exam, whether it's this class or any other class. So we can talk about those tips again as we get closer to the exam. But yeah, you don't have to be too stressed about it. I hope everybody will get A's. That would be great. Usually the average scores that I get on the exams are in the mid to high 80s. So that's for the average for the whole class. So there usually are one or two students who score 100%. So hopefully there's more this class. So it's definitely possible. And with the exam, I mean, since we're talking about it, generally I like to write the questions. So most of the questions are kind of a reasonable difficulty. And then there's maybe 10 or 20 really easy questions, and then maybe 10 or 20 pretty difficult questions. So that means things that you really have to understand the concepts to be able to answer those well. So most of the test is kind of reasonably difficult questions. If you go through the questions at like the end of chapters, similar to those. And then some that are really easy, kind of just throwaway questions. Like if you were even half paying attention, you would definitely get those right. And then a small number that's pretty difficult. All right, let's get jumping into chapter 2. Are there any other questions about exams, logistics, study tips, things like that? All right, well, let's jump into chapter 2 then. So with the beginning of chapter 2, we're going to be talking a lot about biology and genetics. So we'll start talking about natural selection and how that's related to evolution. So natural selection is when the individuals of a species that are best adapted to their environment, they're more likely to survive and then reproduce. And so that's essentially what's determined a lot of the genes that animals, that humans have to start off with. So here's some example of sketches of beaks. So different kinds of seeds or eating different things will lead a bird to have a different kind of beak as it evolves. So if one family of birds splits to 2 different islands and they're eating different foods over the course of a couple 100,000 years, they might look dramatically different because of how they've had to adapt to their environments. So with that, these adaptations come from genetic mutations. So similar to like the X-Men, we don't get, unfortunately, like lightning powers or powers to control the weather. You might possibly have the ability to heal certain kinds of wounds a little bit faster, but not to the level of like Wolverine. But those little bit of genetic mutations can add up. So as those are passed on and become more common in a population, it becomes the norm to have whatever that genetic mutation was. So different colored skin, different length of fingers, things like that. We could get a mutation to have an additional finger if it was adaptive. Probably wouldn't be though. Maybe I could see it could possibly be adaptive to maybe like lose fingers, like lose a pinky or something like that, if people were getting injured and dying more easily because of that, just as a silly example. All right, so with that being said, evolutionary psychology looks at that, how genes are passed down through generations and how that might have shaped human behavior over time. So how did it shape human behavior to come into groups and to join together, to work together rather than working as individuals? How has it shaped how people do things like hunt, find shelter, raise young people? So people coming together and being able to be in groups allowed us the ability to have children that were able to be younger or have a longer developmental period. So if you look at, say, cats or a lot of other mammals, you'll see that their very young stage is very quick. So they're very helpless for quite a short time as compared to humans. So I couldn't take my 4-year-old and just like let them loose in the wild and expect them to fend for themselves. So it's many, many years until a human's able to sustain themselves. So they need support from others. So a lot of that has come from evolution and the genes that have passed down, the mutations that came, brain size developing, things like that. So with that, some of these genetic mutations and adaptations are really, really good for us today. Some are not so good for us. So as an example, our ancestors lived in a food scarce environment. So we evolved the desire to crave high calorie and high fat foods. So when it was more difficult to get those, it was good because then that human would eat the high calorie, high fat food. But in places where you can more cheaply get, say, a McDonald's hamburger than maybe a salad, it's not as good because then we tend to eat a little more than we maybe should. And so that's one thing that in our genes might lead us to have more obesity. So even though these might have been adaptive in the past, in our current environment, some of these adaptations might not be so adaptive. Another good example is our stress responses. So our stress responses in the past were more focused on more immediate dangers like fight or flight in an immediate situation. You might have seen the video, I think it was like 4 months ago now or something, with the mountain lion following the person down. I think it was in Provo Canyon. So, I mean, that was kind of a long video, a long interaction, but it was still only like 10 minutes or so. So that fight or flight situation and that high stress, that's literally what we've evolved to get through. So we haven't evolved the capacity to handle the stress of there's an exam coming in a couple weeks and a final exam coming in a couple weeks and all these other stresses in our life. Our car is going to need maintenance and we need to budget for that, those kinds of things. So you might start feeling anxiety as I say those things because these evolved adaptive stress responses are being activated, but we've evolved for these more immediate things like there's a mountain lion here, we need to get away or we need to kill the mountain lion. And then the stress stops. So that's what we've evolved to do, is have a stress, get through it, and have that stress stop and be able to relax. But in our current environment, we don't really get that opportunity with some of our stresses. So another example of something that has been adaptive to humans in the past now is no longer necessarily adaptive. So an evolutionary psychologist would look at that and say, oh, this is maybe why people are behaving in this way or getting anxiety disorders, because we evolved these capacities and now those capacities are working in this way in this current situation, and it's not as good for us as what we evolved to get away from mountain lions. So with that, evolution alone doesn't dictate our behavior. So while we have this evolved capacity to react to stressful situations in that specific way, it doesn't mean that we can't change our behavior or our choices to make the environment different or make our responses different. So we have these evolutionary capacities that we've built over generations, but they don't dictate 100% of our behavior. So with that being said, though, let's talk about genes. What do genes do? How do they work? And we're doing a really, really brief overview of this. So if you've taken a biology class at the college level, you've probably gotten a lot more detail than we're going into today on how genes work. And so that's great. And so you should be ready and have heard a lot of these things. So we'll start with chromosomes. A chromosome is a thread-like structure in every human cell made of DNA. So inside the nucleus, you've got a bunch of chromosomes, and it's got the DNA. It's basically the DNA wrapped around itself. So the DNA itself is a long molecule, so a really long strand, a really long molecule, and with that, it contains genetic information. So what is a gene? What is that genetic information? Essentially, it tells cells how to develop and what proteins to create. Proteins can come and read, basically grab onto a part of that DNA and see, oh, I should go and do this job in the cell. So that's essentially what's happening here. You've got the chromosome, which is made of wrapped up DNA, and then a protein can come and read a section of that and see, oh, I should go and do this job in the cell. I should go transport this chemical from here to here, or something like that. So it's as simple as, well, as simple as that. That's about as simple as we're going to go with it. And then so what those proteins do, they're essentially the things that build the cells. They direct and regulate the processes. So what genes do is, the genes are kind of like a blueprint. They're kind of like directions to tell the cell like how to work, how to live. In the beginning, as a cell develops, these genes tell the proteins, what kind of cell am I going to be? Am I going to be a neuron? Am I going to be a hair cell? Am I going to be a skin cell? Am I going to be a liver cell? So that's another thing that these genes do. So those mutations I was talking about, they usually happen at this level of the DNA. And so those are the things that can lead to the cells doing different things, us having maybe a little bit better ability to say, maybe some more white blood cells or something like that. That helps us fight infection better. And if that's the case, then that would more likely be passed on to future generations. So that happens at the level of DNA. And so that's what the mutations are. And again, the DNA is being read by the proteins and kind of directing what a cell is supposed to do. Kind of slide out over here away from the sun shining straight in my eye. So what those genes do is they work with other genes and with other non-genetic factors to create and direct proteins to do things in cells. That's about, I mean, it's a lot, there's a lot of complexity to this. You could take entire courses on just the process of how proteins and DNA work to direct and build cells. But just know it's kind of just different chemicals in your body working together to direct how the cells work and function and live and move and transfer energy around them. So with that being said, things like stress, things like exercise, nutrition, radiation, temperature, sleep, they can all affect this reading of the DNA and then expression of those genes. So depending on if you're getting proper nutrition, the proteins might not read this in the same way as they would if you were getting proper nutrition. So they might direct the cell to do something slightly different than it would have otherwise. So that is called epigenetics, and we'll talk about that in a little more detail in just a second. So as researchers look at genetics and try to figure out, well, which of our behaviors, which of our traits are dependent on genes versus what's dependent on the environment, that nature-nurture thing, right? So what comes from the nature of those genes versus what comes from the nurture of the environment, we oftentimes use twin studies and adoption studies. So what would you be doing from your reading? What would you be doing in a twin study versus what would you be doing in an adoption study? What might you be looking at and comparing there? Either raise your hand and then I can see it come up, or feel free to type your answer as well. Or maybe another way of phrasing it, what would you learn about genetics, the nature versus nurture, from a twin study? All right, we've got one. Yeah, go for it.
Okay. So I'm a twin, and even though like I think people wants to see like what's similar between the twins, and we are fraternal twins. Since we grew up in the same environment, had the same parents, even though we had all that, we're still different. And some things that I would choose, she wouldn't. So I think that's kind of what they're looking for. And the funny thing is too that some things that, like right now she's married and she's living in Denver, but like she got pregnant like last year, and I felt all the and like sickness, and I was throwing up, and I couldn't smell anything. I couldn't go to the grocery store, and she was just fine. She had nothing. And when she was ready to give birth, I was feeling the pain on my pelvic area. And when she started breastfeeding, like I was feeling pain on my breast. And she was fine all the time, and I was feeling like all the pain.
That's
which
interesting.
Is, yeah. So I think there's a lot of things to study between twins.
Yeah, absolutely. And it's interesting too. You're talking about feeling similar things that you might expect her to feel as she was pregnant and giving birth. Oftentimes you see that in partners of a pregnant person, that they will often have kind of I think it's called sympathetic pregnancy. And so some similar things. And so usually that happens when people are really close to each other. Oftentimes they're also close in like distance, like living together. But I don't think I've heard of that from people living further away. So that would be interesting, maybe looking up sympathetic pregnancy for a question paper, if that's something you'd be interested in looking into and seeing if that's more common in, say, twins versus other people. Because oftentimes it's like a close relationship and people talking a lot to each other. So I'm assuming that might be what's happening with you and your twin as well. But yeah, really great examples there, especially related to twin studies. Oftentimes that's exactly what people are looking for, is what are the things that are similar between you and your twin versus what's different between you and your twin. Those things that are similar, well, there might be a genetic component to that because you share genes. Whether you're identical or fraternal twins, you share either all your genes or many of your genes. So with that being said, those things that are the same, well, that might be a genetic cause, or primarily genetically caused. Those things that are different, then we can know, well, that's probably not genetically caused, or at least not entirely genetically caused, because they're different between the twins. So really great examples. Can you think of maybe one specific example of something that's the same between the two of you, even if it's just like hair color or something like that, and then one thing that's different? It can be psychological or physical, if you'd be willing to share that.
Sure. Well, we always had different tastes for guys. That's a good thing, I guess. And like the guys that I like, she likes the handyman kind of type, and I'm more like kind of the nerdy kind of type. So that's different. And things that are similar. Actually, we are identical twins, not fraternal. I don't know why I said that. So we are very similar. We are like mirror image of each other. So I have like a crooked tooth towards to the right. So she has the same crooked tooth towards the left. And the funny thing is, like, when we were growing up, my tooth was perfect. But she didn't want to take her tooth out, and I think that caused the tooth to get crooked. So once her tooth started getting crooked, mine started getting crooked too. And it was just right before. So
interesting. Really good examples too. Yeah, thank you very much for sharing. I don't often have a twin in the classes to give such direct examples, so thank you. That's great. The tooth thing definitely sounds like it's primarily genetically caused, so that something in the genes led to your twin having that happen earlier, and then for you it happened just a little bit later, versus tasting guys seems like it's probably not genetically caused, since there's such a big difference there. So really interesting. So some of those things sound like more nature side, some sound more nurture, but like we said at the beginning of the class, there's probably some component, even with tasting guys, that there could possibly even be some kind of genetic component there, but it's obviously not the primary component, as an example. So really great examples. Thank you very much. Adoption studies are also kind of looking for the same thing. They look for a child in their biological family's home versus the adopted home, what things are different between the adoptive parents and the biological parents and the child. So are there traits that they're showing that are more similar to the adoptive parents? That might indicate that those things come more from the nurture side of it. Versus, are there traits, even though they're in a new environment, are they still showing certain traits or interests that are similar to their biological parents? That might suggest that those things are more genetics. So kind of towards that nature side. So these are some of the ways that we can look and try to identify that. And it's really the only way that could be used until we had better genetic modeling technology, being able to figure out a person's genes and genomes. And now we can do something like 23andMe and find a lot of our genetic tendencies. So that wasn't possible up until recently. So that's been really nice to try and better narrow down what comes from genes, what comes from the environment, what is the combination between those. So, oh, wrong screen. All right, so with that, do remember that correlation does not mean causation. So just because something is related to one another, so just because most twins have such and such phenomena that's similar between them. So let's say, for example, let's say if one twin has suffers from depression in their life, let's say, I'm just making up a statistic, I don't even know if this is true. Let's say if one twin suffers from depression, 90% of the time their twin also suffers from depression. Just because there might be that relationship doesn't necessarily mean that there's a causal relationship there, that the genes do definitely cause that, because there's other factors, environment, things like that. So we can't just assume that because there's a relationship that there's a causal relationship. So just as a reminder. So especially with these heredity environment correlations. So as well as some things coming from the genes, that nature side, a lot of things come from the nurture side, from the environment. So we'll talk about a couple of those examples, and then we'll get into one that I think is the most fascinating is epigenetics. So a passive genotype environment, this is essentially when the environment is already supportive of the genes an individual has. And we'll talk about this in the framing of a child. So the parents, the biological parents of a child, they probably have really similar genetics to that child. So let's say, this is probably not actually a genetic trait, but let's just use that as an example. Let's say the parents like to read, or are faster readers, or have better reading comprehension genetically. So those probably aren't actually genetic, but just as an example. So let's say that is genetic, just genetically they're better readers. Well, probably they will have an environment, a house or something with more books. Maybe they're more likely to give books earlier on or read more books at bedtime, something like that. So that is a passive genotype environment correlation. It's already supportive of the genes that this child has. So, but the child didn't do anything to make it good for their genes. So evocative genotype environment correlations are when the child's characteristics elicit a specific environment. So they're not like choosing or actively changing their environment, but because of the behavior of the child, their environment will change. So let's say a child is more genetically inclined to be friendly and outgoing. So if they are, probably they will elicit an environment around them where they have more friends, versus if genetically they're a little more uncooperative or maybe more introverted, they might not make as big of a friend group. So they are not actively choosing to necessarily go out and try and make more friends in this example, but because of their genetic tendencies, they elicit an environment that's supportive of those genetic tendencies. So that one's evocative. So it kind of evokes. Because of who they are, it changes their environment around them because of their behaviors. So the next one is very similar, but different in the key way, in that it's active genotype environment correlations, or it's also called niche peaking. Niche. Never mind. Just read the words. I'm having troubles saying that one right now. But essentially, in this case, the child has a genetic tendency, and then they actively seek out an environment that's supportive of that tendency. So if, again, reading was genetic, or really fast, or reading comprehension was genetic, they might seek out more opportunities to read. They might go to more libraries. They might take more courses in school about language and literature. An outgoing child might look for more opportunities to be outgoing. They might join more clubs. So this is them actively choosing different things. So because of their actions, it now changes their environment in a way that's supportive of their genes. So again, we can have the passive ones where the environment is kind of already set to help the child's genetic tendencies get boosted. Evocative, where because of their behavior, their environment changes around them. And then active, where they are choosing and selecting things because of their own ingrained genetic tendencies. So again, this is one way that our genes interact with the environment. So we have the genetics, and then we can, for active, we can select specific environments that support those things. And so it will boost those genetics. If we don't have the opportunity to pick that specific niche there, then we might not have those genes. We might not see the full outcome of what those genes could lead us to. So that's one example of nature and nurture working together, or I guess a couple examples of nature and nurture working together. One that I think is the most amazing is actually epigenetics. And so this is when the environment actually influences how those genes are expressed. So this, just to be clear, this doesn't actually change the DNA. Your DNA always stays the same unless it's like mutated from radiation or something like that. But the proteins reading the DNA, they can read it differently based on environmental factors. So even though the gene, the strand of DNA, the molecule itself stays exactly the same, and if you cloned a person or you had a twin that was identical, that gene would still be exactly the same. Because of your environment, because of the things that you're doing, maybe your nutrition, your sleep schedule, the proteins might read those genes differently and then make cells do slightly different things. So we'll talk about a couple specific examples of that. So again, a baby will inherit genes. An example of epigenetics is the prenatal development. Do they have toxins, or is it a healthy environment for their development? And then some genes might activate or deactivate or activate at a different range or level based on the environment in the prenatal development. And then they might have different physical features, different mental capacities. The neurons might be a little slower to myelinate, things like that, which will then change a lot about behavior down the line. So again, epigenetics is mostly about the changing of the reading of the DNA strand. So with that, the DNA strand, while the order of it doesn't change, so it's like ATCG, ATCG. So while that order doesn't change, it can get squished together or kind of stretched out in a way that makes that protein read it differently. So because of that change in the twist in the molecular structure, so the literal like structure of these, let's say all my fingertips are a different chemical. So ATCG, ATCG. So if these get too squished together, the protein might read these as one of them, but not all of them, so then express the gene slightly different than the genetic code actually has written it for. So this is another example of bidirectional interaction. So as well as an individual interacting with their parents, with their school network, with their community, our environment can also interact with the way our genes are read and expressed, which is pretty incredible. So I've said that quite a few times. It might be hard to understand without specific examples. Your book gives a couple examples, but I actually really like this video to explain it. It's got a couple really good examples, and I think it's done really well. So let's watch that. Feel free to post up any comments or questions in the comments area here of the class, so I can get to that as we're watching this video. Because I know epigenetics is a really new idea, probably for most of you. So if you're confused, if you still don't understand anything, I'm trying to keep it simple rather than go too complex, but sometimes it's easier to understand it if you know a little bit more of the complexity, then it'll connect with other things you know in, say, biology or cellular development. So let's watch this on epigenetics.
So say you have a clone or a secret identical twin, because you know that a twin or a clone is a genetically identical person to you, which is crazy, but it's true. Anyway, so you have this clone or your twin or what have you, and your parents decide that it's too much trouble to keep both of you. And so they give your clone to a kind of sketchy traveling circus. Now, I know your parents probably wouldn't actually do this, but just roll with me for a minute. So for the purposes of this example, let's just say that you grew up living a normal suburban American life, while your clone grew up with less stability and less access to nutritious food and less education, but more exercise and more access to sword swallowers and bearded ladies. And let's say you guys are 50 years old or whatever, you get together and have some lunch. So you're sitting there across the table from your clone. What do you think you see? Well, for starters, Clone probably has a tattoo on its face. And if Clone had stayed in the circus, Clone probably, professionally rides a unicycle over a tightrope, so Clone is probably in pretty good shape. But then again, Clone probably smoked cigarettes and probably was malnourished as a child, so might be slightly shorter than you. By the same token, let's say that you've been eating Sonic tater tots 5 days a week since 1992, so you're looking a little bit tubby. Plus, you spent the last 20 years at a really stressful job that puts you at risk for all kinds of weird health complications. So, at 50, you and Clone would probably look pretty different from one another. But it turns out, you don't just look different, you actually have become different. Now, if some scientists looked at your genetic code, they would say that your DNA was still exactly the same as that other person. That is to say, if your genome is a paragraph, all of the letters would be in the exact same order. But on another level, there would be a huge difference. And if we extend this metaphor, we can say that the letters are in the same order, but the spaces and the punctuation are all in different places. Of course, completely, potentially changing the message of that paragraph. The study of this genetic punctuation that I'm talking about is called epigenetics, which literally means above genetics. The epigenome doesn't change your DNA, but it decides how much or whether some genes are expressed in different cells in your body. Epigenetics looks at what happens to your genes over the course of your life and whether those changes could be passed down to your children or even your grandchildren. So here's the way epigenetics works. You have billions of cells in your body, and they each contain your DNA, the same exact blueprint of your genetic code. But just because they have the DNA doesn't mean that they know what to do with it. They need outside instruction from these little carbon and hydrogen compounds called methyl groups. The way these methyl groups control the genome is by binding to a gene and saying, do not express this gene. The methyl groups bind differently to your genome in a skin cell versus, say, a tongue cell or an eyeball cell. And that is one of the ways that a cell knows that, hey, I'm a skin cell, or hey, I'm an eyeball cell, or hey, I'm a muscle cell. In addition to methyl groups, epigenetics is also controlled by histones, which are proteins that are basically the spools that DNA winds itself around. Histones can change how tightly or loosely the DNA is wound around them. If they're more loosely wound, the genes can express more, and if they're more tightly wound, then they express less. So whereas the methyl groups are more like a switch, the histones are more like a knob. Every cell in your body has a distinct methylation and histone pattern. And that is what gives every cell its marching orders. Think of your genome, the DNA, as the actual hardware of the computer, while the epigenome is more like the software, which tells the hardware what to do. The genome is what's going to be doing all the work, but the epigenome is going to be telling it what to do. So the hardware of your DNA is going to be the same throughout your entire life. But these epigenetic tags do change throughout your life, and they decide what genes get expressed or not. Now, epigenetic information in a cell isn't permanent. It can change throughout your life, and it can be hereditary, and it can change over time, especially when your body is going through a lot of changes, like, say, during puberty. A bunch of methyl groups kick in and they're like, okay, so you guys over here, you're gonna have to start growing hair, and you guys over here, I really need you guys to get behind giving this guy some really horrible acne. Or like when you get pregnant, which hopefully I won't, but when you do, your epigenome has to be like, all right, guys, everything's about to get a whole lot bigger in here, and we're going to have to pass something the size of like a Merriam-Webster's dictionary through that little pipe over there, so let's get this thing done. But it's not just these dramatic times when the epigenome is changing. It changes subtly throughout our entire lives, and it changes based on a lot of environmental factors, like what we do, what we eat, what we smoke, and how stressed out we are on a daily basis. Scientists have found that things like a bad diet can actually lead to methyl groups binding to the wrong place and making mistakes. And with those bad instructions, cells become abnormal and become a disease. And then basically all hell breaks loose and you get cancer or something. Epigenetics is a very young science, though we've known about the epigenome since the 1970s. It's only in the last 20 years that we've even known what effect these epigenetic tags are having on our DNA. And even after they got all that business straight, scientists still thought that all of our epigenetic tags were stripped off of our genome before they were passed on to our children. So if you started smoking 10 packs a day when you were 10 years old, that would certainly be a horrible health decision for you, but you wouldn't necessarily be harming your unborn children in any measurable way. However, the thinking on that is changing pretty rapidly, because it's true that a lot, even most of the epigenetic information from a parent is stripped off of the embryo's genome in the first few days, and fresh ones are created specifically for this new person. However, some of these tags get stuck on the genome and are passed down from generation to generation. And it just so happens that the more they study this, the more it looks like bad epigenetic information is being passed from generation to generation. And this is a whole new way to think about how we pass information between generations. Your grandmother was making dietary decisions that affect you today. As we experience all of these new strange epidemics, diabetes, autoimmune disorders, cancers that weren't appearing in previous generations, it's starting to look like these may be caused by epigenetic information passed down from our parents. I know, it's such an unbelievable buzzkill. There is no point in our lives when we can do anything without guilt anymore. The discovery that the environmental factors of parent experiences can be passed down from generation to generation was sparked in the 1980s. And this happened when some scientists were looking at the birth and death records of some people who lived in 19th century Sweden. As is a weird place to find a genetic revolution, but there it is. It wasn't just any place in Sweden. It was in Norrbotten, which is the northernmost county in Sweden, which is literally in the Arctic Circle. And despite the fact that Norrbotten was literally the worst place you could possibly choose to live in Sweden, there were some people living there in the 1800s. And they were completely cut off from the rest of the world. Let me clarify. These people were isolated. Like, if they didn't have a good crop year, people died. No, it wasn't ideal, but, you know, they didn't ask me my opinion, so I'm not giving it. So anyway, these people subsisted entirely on what they grew and the animals that they raised. And like I said, sometimes they starved, but sometimes they had huge, bountiful years of plenty. And when it happened, people totally went ape crackers. I mean, of course they did, because they were so freaking hungry, and then all of a sudden there was all this damn food everywhere. Anyway, there was a public health specialist who was looking at the effects of the people who grew up in the really bad starving periods of time versus the people who grew up in the eat all you can at the smorgasbord years. And you might already be guessing what they found out. People who went from relatively slim pickings to feed your face until you have to barf and then do it all over again in a single season, those people died an average of 6 years sooner than their starved out counterparts. And you know what sucks? So do their kids. And so do their kids' kids. Boo epigenetics. So, of course, now we all know, and we're all going to stop doing unhealthy things starting today. Unless the damage is already done. The damage is almost certainly already done. But hey, epigenetics brings good tidings along with the bad ones. For instance, we now know that certain types of cancer are caused by misplaced epigenetic tags, and scientists are now developing drugs that can silence the bad genes that were supposed to be turned off in the first place. Additionally, until recently, we thought that genes were the end-all and be-all of who you got to be. They were your blueprint, and you couldn't escape them. This outlook is not just kind of depressing, it also leads to a yucky sort of social prejudice. Because when you look at data without considering social and epigenetic factors, it might look like people with less money are less intelligent. So just like 15 years ago, there were scientists saying things in public, mind you, like, some people just have good genes for intelligence, and it just so happens that the poor people don't. Oh, it's so sad. Too bad, poor people. But you have bad genes. Well, it turns out that that is not even a little bit true. Not only are there a huge variety of social factors that affect how well people do on intelligence tests, but a genetic trait is also not just a product of genes, it's also a product of environment. Any one person's genome is determined by any number of decisions made by any number of their ancestors. And right now, you are making decisions that are going to affect people who are alive long after you're dead. No pressure or anything. And so I'm glad you've watched this, because when you and your clone are in your 60s and you sit down, have lunch, and he brings his son and his son has a tattoo on his face, he did not inherit that from you. This is Hank Green for The SciShow. We hope you learned something.
So hopefully that makes the explanations a lot more clear. This other video, it's kind of interesting because it talks about epigenetics in relation to Pokemon, which is kind of fun. But there's a couple really good examples of both human and animal features that are caused by epigenetics. So let's watch a little bit of this to get some more examples. So hopefully it helps you understand it a little bit better.
Epigenetics. Epigenetics is something that you don't usually get into unless you're majoring in this stuff in college. So all you younger viewers, get ready to blow your teachers' minds when you drop this knowledge bomb in the middle of your lesson about Punnett squares. Those are so like 4 years ago on this show. So what is epigenetics? At its core, it's essentially the phenomena by which animals are born with one set of genes, but potentially have parts of their DNA that are locked at birth and can later be unlocked by certain things happening in the environment. If that sounds weird, think of it this way. You have a ton of DNA in your body, 2.9 billion base pairs, but not all that DNA is working all at the same time. Without getting into the nitty-gritty of genetics, your cells have specialized proteins that turn off and on different genes throughout the course of your lifetime. When you're a kid, you have a
So in the last video, we were hearing that's the methyl groups and the histone patterns. So that is what is either turning the genes on and off or making them be read in slightly different ways. So again, that's literally the structure of the DNA being read differently depending on your environmental factors. So if you're having good nutrition, your DNA might be read in one way versus not read in another way.
Ton of genes turned on to do things like grow your bones, build your brain cells, and create the hormones that make you fall in love with literally every creature that you pass in the school hallway. Later in your life, though, some of those genes get locked back up. When that happens, maybe your metabolism starts slowing down. You stop producing color for your hair, and it turns gray. As you get older, you have all the same genes that you started with on the day you were born, but how you look is depending on which ones are turned on and off. Your epigenetics. Right now, my baby Oliver has blonde hair, but Steph and I have brown hair. So, should I be worried about Steph's relationship with the milkman? Is Steph cheating on me? Of course not. At least, I don't think so. Steph! Steph, are you cheating on me? Oh, yeah, sure, that's what you would say. Now, what's really going on here is that right now, most of the genes in Oliver's system that produce melanin, which turns your hair brown, are turned off. Like, way off. Just like I had when I was his age. But when he gets older, those genes get activated, and his hair will turn brown just like ours. His physical traits will change as his genes turn on and off. Now, with animals, what epigenetic researchers have discovered is that some animals will be born with one set of genes, but potentially have parts of their DNA that can be unlocked by certain triggers in their environment. A great example of this are honeybees. Yeah, I know. I just dedicated an entire episode to bee talk as it relates to pt1. Maybe I'm just going through bee movie meme withdrawals. I don't know. Bees are fascinating creatures, and in this case, they are the perfect EV example. So, like I covered back in that episode, female bee larvae can grow into either a queen bee or a worker bee. What makes the difference here is the food that they're given at birth. Bees given royal jelly become a queen, while everyone else becomes a worker. So in this case, royal jelly is basically the evolutionary stone for bees, determining what form the larval bee evolves into. It even changes the bee physically, where, in their adult stage, queen bees and worker bees look way different.
So with that example, hopefully you're understanding then as, oh, come on. There we go. That's the picture I wanted up. So this bee and this bee have the same, or really similar, because they're essentially siblings, have very similar genetic codes, but they look very different. If you cloned this one bee, this queen bee, and then you gave one of the clones the royal jelly in the larval stage versus other foods in the larval stage, they would both turn out looking very dramatically different. So physical structures, their behavior is different, and that's because of that diet, the genes are read in a specific way to activate certain aspects of the genes for the queen that are not active for the worker, and vice versa. So let's look at one other example that I think has a really good, might give you a good sense of the range that you might find. Where is it? All right, it's the next example talked about. Good. So we'll watch through that about mice.
Bees and worker bees look way different, but in their larval stages, they're practically identical. It's exactly like two different evolutions that wind up with two different appearances despite starting with the same looking EVs. The queen and worker bees didn't start off as genetically different, but differences in their environment, the royal jelly, or lack thereof, led to wildly different gene expressions. And bees aren't the only place in nature where this happens. Another EV-like example happens in the agouti mouse, which has a gene which changes the fur of its coat. These two mice are genetically very similar at birth, but due to differences in nutrition, they have very different gene expressions, with one having a gene expression that causes it to become obese and have lighter fur. Same DNA sequence, different phenotypes, all due to environmental factors. In humans,
So it goes on to talk about the example of diabetes in humans and how humans might,
but due to differences in nutrition,
and that humans might have diabetes if they have the genes, and then they have a diet that will then lead to diabetes. This picture, I think, is a really good example, though, of the genes being at different levels. So that knob we were talking about, so with the the tightness or looseness of how the molecule of DNA is wound will lead to the genes being expressed slightly different. So you've got a very brown mouse here that's very small versus here. These are all siblings essentially from like the same litter of mice. So genetically, they're very similar mice. You could do this with mice clones that are identical genetically. That just costs more, so they just use siblings. But you can see here this range of coat colors and the size of the mice as well, and that's based just on the amount of like calories and fats that they were getting in their nutrition. So a lot of human genes might be read or activated differently or more or less based on environmental factors. So that's what epigenetics is all about, is that we have these genetic tendencies, but with that, our environment, even our own choices, might change how those genes are expressed. So again, the DNA doesn't change. The DNA is still the same. If you cloned yourself at 2 versus 40 versus 80, those clones would all be genetically the exact same as each other, but the epigenetic information on in those clones might be different at those different ages. All right, so this gives us another thing to look at with those developmental cascades. Oh, we've got a comment here. Yeah, so it will be interesting to learn why some genes adapt while others don't. So hair color changing versus eye color that doesn't. Yeah, so if I'm not mistaken, I believe eye color can actually change very early on in life, but after I think it doesn't change after about age 2, but it can also change if I'm not mistaken. Because it's the colored part of your eye is a muscle, so those muscle cells, if they have different pigments in them, you'll have a slightly different colored eye. With that being said, though, this goes back to our talking of developmental cascades. So you not only have the genes that might suggest that you'd be over here for any kind of outcome. Maybe over here is not getting diabetes and over here is getting diabetes. So genetically, you might be very unlikely, but based on environmental factors makes you a little more likely or something like that. Maybe diabetes is not a really good example. But for any kind of behavioral, psychological, physical factor, not every single thing can be affected by epigenetics, but many of our genes can be. So some of our behaviors might come partially from the genetics and partially from the epigenetic factors, and then again, partially from the environment. So there's this whole combination of variables that leads to our genetics leading to different behaviors, which is really fascinating. Yeah, so let's jump to, well, maybe we should take a quick break. That's about all we'll cover for epigenetics. Again, we did kind of skim over what is chemically and physically happening at the level of those proteins reading the DNA, but that's okay for our purposes for this class. If you want to go into it more, it might be a good idea for a question paper to look into it. Some of those research articles might be a little beyond your understanding in some of the sections, but if you read like the intros and the discussions to them, hopefully those parts will be understood. But a lot of times, for any article really, I don't recommend a beginning psych student read through the methods and results of a study. But that being said, let's take a quick break and come back here at 9:30, and then we'll jump into prenatal development and learn a lot about the average development during the prenatal period. So be back here at 9:30. All right, let's get back to it. Let me know if you can't hear me, if the audio did anything funny when I was gone. But let's talk about prenatal development. So we'll start with the different periods. So germinal period is essentially the first 2 weeks after conception. So usually at this point, the pregnant person doesn't know that they're pregnant. So it's just those first 2 weeks. It's when that egg is fertilized, which then is now called a zygote. Then there's cell division as the multicellular organism, that zygote, goes and attaches to the uterine wall. So that period from that fertilization to attaching to the uterine wall, that is called the germinal period. The embryonic period is from that point, that second week, to 8 weeks after conception. So what is happening here is now you have an embryo. You start to see organs beginning to develop. You start to see things like the brain beginning to develop, stomach, eyes, things like that. And here in this period, because so many of these organs are beginning to develop, it's the embryo is now very susceptible to the environment. So teratogens, hopefully you remember what teratogens are from your book. We'll talk about it in a couple minutes. So things like toxins in the environment it's developing in. So these are basically screenshots from your book that go into more detail about what's happening, like breaking down that embryonic period into further periods. But we'll just go with this more broad, time. And then the fetal period starts at about 2 months and lasts till the end of pregnancy, till that baby is born. So it starts from 2 months, lasts to about 7 months. So there's a lot that is happening here. So again, there's a lot of danger of teratogens in the embryonic period, so that first 8 weeks, and then after that, some of those toxins aren't necessarily going to be as bad, but it just depends on the toxin and the time for these teratogens. But there's a lot of things happening here. So as you can see, your book breaks this down into even further categories. So this can be fun if you've ever had a baby or are pregnant, looking at those, seeing what might be happening at different times. My brother is actually, he and his spouse are actually going to have a baby in a couple months, so nearing the end of the pregnancy. So they often will mention, oh, we saw on our app, like these are the things happening. So it's pretty neat to see. With that being said, I wanted to focus here on the development of the brain. Other physical capacities of the body definitely affect psychology and how we behave and think and process information, but the brain is one of the most important organs for that whole process. So let's look at what we can see is happening to the brain during this time. So this is at 29 days, I believe. It's really hard to see in this picture. I've tried to find a higher quality picture or a better one, but I like how many time points this one shows. So this is the brain and spinal cord, the base of the hindbrain here at the back. So 30 days, you've got 52 and 59, and then up here to 9 months. So you can watch and track that time of when it is in the germinal period versus the embryonic period versus the fetal period, and what kind of brain structures you can start to see. So here, it doesn't really look too much like what you might expect from a human brain yet. A lot of cells are still being created and moved, and that's still happening up to about age of 2. So at about week 5,